Biochemistry 401 Lecture 23. Today we're going to start a discussion of glycogen metabolism. We're going to begin with glycogen degradation. This is the mobilization of glucose to fit the needs of the cell itself, and in some cases, the needs of the body as a whole. We're then going to talk about glycogen synthesis. This is the formation of the glucose storage unit. So let's get started. So what is glycogen? Well, glycogen is a highly branched polymer of glucose that's found in organisms such as animals and also bacteria. It consists of glucose units that are attached through two types of glycosidic linkages. In the straight chains, glucose units are attached through alpha-1,4 linkages, whereas branch points are formed through alpha-1,6 linkages, and branch points are found for about every 10 units in the polymer. Glycogen also includes an initiator primer molecule, and this is called glycogenin. This is shown by a yellow circle with the letter G in it. This polymer has directionality, such that the reducing ends are attached to glycogenin and the beginning of branches, and the non-reducing ends are found at the ends of branches. And so, when glycogen is mobilized, this mobilization occurs at the non-reducing ends, and additional monomers are added to the non-reducing ends. Now, the primary storage sites for glycogen are the skeletal muscle and the liver. Skeletal muscle cells contain glycogen that's used for their own needs. The glycogen stores in muscle cells are approximately 2% by weight. However, liver cells contain glycogen that's used not just for the needs of the liver cell, but for the needs of the body as a whole. And so the percentage of glycogen per cell by weight is higher, 10%. We have more glycogen that's stored in our muscles than we do that's stored in our liver. This is because we have more muscle cells than we have liver cells. But for each individual cell of the muscles, we have less glycogen stored per cell than we have stored per cell in the liver. 2% in individual muscle cell versus 10% per individual liver cell. Glycogen is stored as dense granules in the cytoplasm, and this is shown in the electron micrograph to the right. We can see glycogen granules deposited in the cytoplasm. The large circle almost in the center of the micrograph is the nucleus, and mitochondria are the little ovals that are seen in the slide. The dense, dark granules that you see that look almost like freckles in the cell is the glycogen. And these cells are actually liver cells. But why use glycogen? Why not just use fatty acids and fats as a prime storage molecule? Well, this is because glucose is the sole fuel for red blood cells, and it is the primary fuel that's used by the brain. When you break down glycogen, you get glucose molecules that can be used by all the tissues in the body. However, if you were to break down fatty acids, you would get acetyl-CoA. And this can't be used by red blood cells, and the brain would really rather not use this. It can use breakdown products from fatty acids if it needs to under starving conditions, but it is more difficult for the brain to do this, and so the primary circulating fuel is glucose, and the primary storage fuel is glycogen. Also, the utilization of glucose can proceed under anaerobic conditions. Also, glycogen has many branch points for a rapid regulated mobilization, and we'll see this in just a minute. And so here we see a schematic representation in Haworth diagram of glycogen. The linear straight chains consist of glucose residues that are linked by alpha-1,4 linkage. Branch points are formed through glycosidic bond between carbon 1 and carbon 6. And so individual branch points are alpha-1,6 linkages rather than alpha-1,4 linkages. The glycogen-free ends of straight-chain polymers are broken down to release glucose 1-phosphate, and the enzyme that catalyzes this reaction is called glycogen phosphorylase. We're breaking down glycogen into individual glucose 1-phosphate units, and we're doing this by adding phosphate. And so this process is called phosphorylasis. It's because we're using phosphate to break a covalent linkage. This is similar to what we see with hydrolysis, only instead of water, we're using a phosphate. The product of this reaction is glucose 1-phosphate and a polymer reduced in length by 1 glucosyl unit. Now, the glucose 1-phosphate that's liberated can be transformed to a glycolytically usable glucose 6-phosphate, and the enzyme that does this is phosphoglucomutase. And again, the names of these enzymes, both of them, really say what they do. Once glucose 6-phosphate is made, it can go on through glycolysis as we saw before. It can be shuttled off to other tissues. In the liver, we can use glucose 6-phosphatase to release free glucose into the bloodstream for use by other tissues. Another thing that we can do with glucose 6-phosphate is to use this product directly in the pentose phosphate pathway. We haven't talked about this pathway yet, but we will shortly. And so again, there are 3 fates of glucose 6-phosphate: glycolysis, the generation of free glucose, and the pentose phosphate pathway. And one of the functions of the pentose phosphate pathway is to make NADPH. Now, the fate of glucose 6-phosphate depends on the cell type and the current metabolic needs of the organism. We're going to take glycogen and use glycogen phosphorylase to release glucose 1-phosphate and a glycogen molecule that's reduced in length by 1 glucosyl unit. We're then going to use phosphoglucomutase to make glucose 6-phosphate. Now, glycogen phosphorylase cleaves glucose through the addition of inorganic phosphate. This molecule can catalyze phosphorylasis of the straight chains to within 4 glucosyl units of a branch point. Once it gets too close to a branch point, it can't function any longer because it just doesn't have enough room to function. This enzyme yields alpha-glucose 1-phosphate monomers. Now, this is an energetically favorable reaction because the released glucose is already phosphorylated. It's also favorable because this intermediate cannot diffuse out of the cell. Once you've made this phosphorylated glucose, you're not going to lose it. You're not going to lose it to the bloodstream if you're a muscle, for instance. Now, the glycogen phosphorylase enzyme is a dimeric enzyme that cleaves the alpha-1,4 glycosidic bond between carbon number 1 and the glycosidic oxygen through phosphate addition, and it uses a cofactor called pyridoxal phosphate. It's important to make sure that this is a phospholytic, not a hydrolytic reaction. We don't want to lose the phosphate to a reaction that is unproductive. And so in order to do this, we must exclude water from this reaction. In this diagram, we see a ribbon diagram of glycogen phosphorylase. This enzyme is a homodimer that is allosterically regulated. One subunit is shown in yellow, and the other one is shown in white. In the ball and stick diagram to the right, we see pyridoxal phosphate with inorganic phosphate and glycogen in the active site. This active site is organized so that it excludes water from the site of the reaction. This makes sure that phosphorylasis occurs and not hydrolysis. The enzyme is a processive enzyme. This means it's able to hold on to the glycogen molecule and cleave 1 glucosyl residue at a time without letting go of the glycogen molecule. This enzyme can cleave many residues without leaving the substrate. This is the glycogen phosphorylase mechanism. Pyridoxal phosphate is a B vitamin derivative. This cofactor is covalently linked to a lysine in the enzyme to make a Schiff-base linkage. This is shown in the diagram in the upper right of the slide. A Schiff-base linkage is simply a double bond between a carbon and a nitrogen. Pyridoxal phosphate acts as an acid-base catalyst. Phosphoglucomutase catalyzes the transformation of glucose 1-phosphate to glucose 6-phosphate for use in glycolysis. In muscle cells, the phosphate keeps glucose in the cell for the muscle's own energy needs. But in the liver and in the kidney cells, the phosphate is removed from carbon number 6 by hydrolysis in a reaction catalyzed by glucose 6-phosphatase. This is the enzyme that we saw during gluconeogenesis, and it's found in the endoplasmic reticulum. This allows free glucose to be released into the bloodstream to meet the metabolic needs of other tissues. Glycogen phosphorylase can't catalyze the breakdown of alpha-1,6 linked glucoses at the branch points. It also cannot get any closer than 4 residues away from this branch point. And so, 2 additional enzyme functions, a transferase and an alpha-1,6 glucosidase, are supplied by the debranching enzyme. Hexokinase in muscles will phosphorylate this glucose to prevent its exit from the cell. So let's take a closer look at this. In the figure on the right, we see a glycogen molecule. Those residues that can be released by glycogen phosphorylase are shown in red. Those that cannot, because they're too close to the alpha-1,6 linkage, are shown in blue. Now the debranching enzyme has 2 functions. One is actually a transferase, and the other one is an alpha-1,6 glucosidase. The transferase portion of the debranching enzyme is going to take those last 3 glucosyl residues and move them to the end of a straight chain. Then the 1,6 glucosidase portion of this same enzyme is going to hydrolyze glucose off. This is going to remove the branch point. But the trouble is, when we hydrolyze glucose from glycogen, we have glucose that is not phosphorylated, and this glucose can leave the cell by diffusion right through the membrane. And so to trap this glucose in the cell, we're going to phosphorylate it using hexokinase. And this is the same enzyme that we saw in glycolysis. And so to recap, glycogen phosphorylase cannot attack glucosyl residues that are any closer than 4 residues away from an alpha-1,6 linkage branch point. And so what happens is another enzyme comes into play that has 2 separate functions, a transferase and an alpha-1,6 glucosidase. The transferase will take those last 3 residues and move them to the end of a linear portion of glycogen. Then those can be cleaved by phosphorylase with no problem. In the meantime, the alpha-1,6 glucosidase is going to hydrolyze off the branch point glucosyl residue. This results in free glucose in the cell, which can be trapped by hexokinase, if necessary. If we're talking about a liver cell, it's just as easy to let this free glucose diffuse right out of the cell without phosphorylating it, because the liver's job is to just release glucose and not necessarily trap it. And again, this slide points out the fact that the transferase and the alpha-1,6 glucosidase functions are both functions of the single enzyme, the debranching enzyme. Here again to recap, alpha-1,6 glucosidase hydrolyzes the alpha-1,6 glycosidic bond at the branch point and releases free glucose. And the product of this reaction is free glucose and glycogen that is shorter by 1 residue. And so now let's talk about glycogen synthesis. As with any other synthesis, we have to activate the reagents that are going to be used in this synthetic reaction. In this case, to activate glucose, we're going to use UTP. And in this case, the activated glucose is UDP glucose. In this figure, we see uridine diphosphate glucose, which is UDP glucose. The glucose in this figure is shown in blue, and the UDP portion is shown in black. So how are we going to activate glucose? Well, we're going to use glucose 1-phosphate and UTP. This will yield UDP glucose plus pyrophosphate, and the reaction will be driven forward by the hydrolysis of pyrophosphate. And so we're going to use UDP glucose to make an alpha-1,4 linkage to add to an already present glycogen chain. And we end up with UDP plus glycogen that is longer by 1 glucosyl residue. The enzyme that catalyzes this reaction is glycogen synthase. And again, this is a very easy name to remember because it says what it does. Glycogen synthase is how we make alpha-1,4 linkages, the straight chain portion of glycogen. The glucosyl unit of UDP glucose is transferred to carbon 4 of the growing chain. And this chain must have at least 4 residues already there for glycogen synthase to be able to use that glycogen chain as a substrate for addition. And so a primer is needed to start a new molecule. This primer is glycogenin. Here we see a cross-section of glycogen. We can see that it is a highly branched polymer of glucose. In the center, we see a protein that is shown in ribbon diagram, and this is glycogenin. It is a dimeric protein with alpha-1,4 linked oligosaccharides already attached. It is used as a primer to initiate the synthesis of a new glycogen molecule. And so how are branches formed? This happens through the action of the branching enzyme that forms an alpha-1,6 linkage by breaking an already existing alpha-1,4 linkage and moving a block of about 7 residues usually, transferring them to a more interior location, and then forming an alpha-1,6 linkage. The block that is moved is transferred from an exterior region to a more interior region that contains 3 or more residues already. This is the end of Lecture 23. Have a great day.